Welcome to the 54th InterScience Conference on Antimicrobial Agents and Chemotherapy, or ICAAC. I'm Michael Schmidt from This Week in Microbiology, and it's my distinct pleasure to welcome Dr. Lee from the University of Illinois at Chicago. Dr. Lei, also from my alma mater at the University of Illinois at Chicago, and Dr. Jasper Chang from the University of Hong Kong. Together, we're going to discuss a really exciting topic here this morning, something that's emerging, and that's probably something that everyone has read about at least once in the popular news media, and that's MERS-CoV, which is the Middle Eastern Respiratory Syndrome coronavirus. Together with the program committee, we have selected some of the science this week that's being presented here at ICAAC, and I thought it would be appropriate that we share with you some of the exciting results that these three investigators have learned. This is truly InterScience, which is in the name of ICAAC. We have an interdisciplinary project where we're literally going from Dr. Lee's bench all the way to Dr. Chang's bedside. We're literally going to follow the development of a drug that's going to be useful, hopefully, for this emerging pathogen. Middle Eastern Respiratory Syndrome contains this coronavirus, MERS-CoV. This emerged in 2012, so it's a relatively new virus, one for which we didn't have medicines in the past. And so this morning we're going to learn how these investigators are first developing new drugs and secondly repurposing old drugs currently available to us to see whether or not it's possible to treat this emerging disease. So welcome to This Week in Microbiology via ASM Live. So how is everyone this morning?
Good.
Good?
Good.
Good?
Thank you for having us here, Professor Schmidt.
Well, you're most welcome. Thank you for traveling halfway around, more than halfway
It's my
around
pleasure.
the world and braving O'Hare Airport as you came to Washington, D.C. So we're going to start with Dr. Lee. We're going to start at your bench. What have you been doing? We all know, we've seen that MERS-CoV is a frightening virus. It's emerged out of the Middle East, and this past summer we were terrified in Chicago because a patient came through O'Hare Airport, which used to be the busiest airport in the world before Atlanta took it from Chicago. And so how'd you get involved in looking for new medicine to treat this virus? So just tell us a little bit about the work in your lab.
Our group, Dr. Michael E. Johnson and us, have been working on SARS coronavirus target-based drug design and discovery for maybe past 6 or 8 years. So when MERS coronavirus came out in 2012, we were curious that our knowledge and discovery from SARS coronavirus enzyme-based drug discovery can translate to MERS coronavirus once. So since we've been working on two main proteases, it's essential for this SARS or MERS coronavirus replication.
So the protease is important to allow the virus to do its dirty deeds.
Yes. So it's very essential. So if we inhibit this either protease, virus cannot replicate. So we target these enzymes.
And we know that protease inhibitors are very useful in our pharmacopoeia that enables us to attack viruses, because we've seen them, of course, in treatment of HIV as well as in the treatment of hepatitis C with the new drugs that are coming out for hep C. So
yes.
Protease inhibitors are, one of those tricks that fortunately humans have in their medicine chest to treat these viruses.
Yes. So there are two proteases in both SARS and MERS. One is main protease called 3CL-Pro. The other one is papain-like protease we call PL-Pro. So we started with PL-Pro of MERS coronavirus. So we overexpressed the protein, and once we got the enzyme, because we have been working with the SARS coronavirus PL-Pro over the years, we had like 4 SARS PL-Pro lead inhibitors. So
you had them in the freezer ready to go.
Yes. So we wanted to see if those SARS PL-Pro inhibitors would work for MERS-PL-Pro.
Because while protease sounds like one protease would be the same and one virus would be the same in the other, you don't know whether the inhibitors are going to be active in that other virus.
Right. We don't know until we actually try. So we tested 4 of them, none of which were effective to MERS coronavirus PL-Pro.
None were.
No.
So is this where Dr. Lay comes in? He begins to tinker?
Not yet.
Not yet. Okay.
So, I mean, it was actually parallel because
Okay.
we need a structure of the APO MERS PL-Pro to see actually how different those SARS PL-Pro and MERS PL-Pro structures are.
So this is really fundamental basic science where you really want to understand how this protease is attacking its substrate. You want to see what's active so that you can then target your inhibitor to the right spot.
Yes. So, how started working on the crystallography to solve APO MERS PL-Pro structure, and I started the high throughput screening with our very special compound library.
So was he a very demanding colleague? He wanted you to make lots of this stuff so he could crystallize it, or was that his job?
I think crystallization is mostly my job.
So you had to make the massive amounts and then make the crystal of this molecule and then the inhibitors?
Actually, we're trying to get the co-crystal structure, but at the very beginning, we just want to know the structure of the protein itself, because we need to know the protein itself first, and then better to know how it interacts with the inhibitors later.
It's sort of like finding a parking spot in the busy city of Chicago. You got to find where that empty spot is and to target your inhibitor.
Yes. So basically, I character myself more as a structural biologist. Hyun wanted to talk to me do you want to we collaborate and start the MERS PL-Pro project? I was like, yeah, of course, I would like to. So at the very beginning, I did a little bit sequence alignment studies, and I think the MERS PL-Pro and the SARS PL-Pro actually shares a similarity above 70%, which is fairly high. We just assumed that since the similarity is so high, and I did a little bit of homogeneous modeling, and I believe the structure similarity between them, the 3-dimensional structure between them should be high too. And also we assume they're gonna have the same inhibitor selectivity, which turns out is totally ineffective of those inhibitors, of the SARS inhibitors against the MERS. So which kind of surprises us at the very beginning. So later on, after I solved the MERS PL-Pro crystal structure, this even shocks us more. It's because after solving the structure, we made a comparison between the SARS and MERS PL-Pro, and we find the overall structure of these two proteins are very similar. And even the catalytic sites, they overlapped very well. So it really doesn't explain at the very beginning how come those 4 inhibitors against the MERS are not effective at all. So later on, after we thoroughly examine the structure a little bit more, we're trying to find the answers from the structure-wise, and we find there are 2 amino acid residues from an important loop that are different from the 2 species, which these 2 amino acid residues in the SARS are forms of chemical interactions with the inhibitors in the SARS, but it replaced by the other 2 different amino acids, which lost the chemical connections with the inhibitors. We believe this is the major reason why those inhibitors are not effective against the MERS.
So Dr. Chan,
Yes, Professor
you're on
Schmitz.
the front line taking care of sick people.
Yes.
And you obviously have seen SARS from the area of the world that you come from.
Yes, that's correct.
SARS emerged out of Asia, and during your clinical career, you probably saw some SARS patients.
Yeah.
And so when you read for the first time about the emergence of MERS, it probably got you very concerned because this was an agent not endemic to Asia, but endemic to the Middle East, and you were probably faced with, my goodness, I'm going to have to take care of a whole new class of virus for which I don't have drugs. So why don't you share with us how your role in this process came about?
Right. So our team has been working on coronavirus research since the time of SARS. So during the post-SARS era, we have been actively surveying different animal species, especially bats, and we found numerous bat coronaviruses and also other mammalian and avian coronaviruses. And together with the human coronavirus HKU1, which is another human coronavirus, we now know exactly where the MERS coronavirus is situated, phylogenetically related to the other coronaviruses. So in fact, at the discovery, we were quite surprised when MERS coronavirus emerged in 2012. We found that the most phylogenetically closely related viruses were the bat viruses, HKU4 and HKU5, which we found in bats in southern China at that time. So we were thinking whether this is, again, another human coronavirus that emerged from bats.
And that's what I think they're learning out of the Middle East as well, is
Exactly.
that there's a bat species that particularly frequents or roosts in tents, if you will, of the nomadic tribes in the Middle East.
Right.
And that may have how it, jumped into the camels and everything as they
That
say.
is the hypothesis that is being taken now. So the important animal reservoirs that people consider nowadays to be related to MERS coronavirus are the camels, dromedary camels, and possibly bats in the Middle East or in Africa. And to clinician scientists like us, we always want to find a cure for the patients.
You need medicines
Yes.
because that's what people want.
Yes.
Doctor, fix me.
Yes, hopefully. And so in any emerging infectious diseases, time is always against us.
Yeah.
And we can see that the outbreak has grown in the past 2 years. Now it's more than at least 800 cases with nearly 300 deaths. So one of the approach to find new drugs is what the excellent work of Dr. Lee and Dr. Lei have done, is to find specific enzyme inhibitors that will inhibit the replication cycle of the virus. But that would take years or at least months before it can be used in
Well,
humans.
you need to test it in humans, you need animal studies.
And so the other approach that we could have adopted was to see what is available in hand. And we can do that with two methods. First is we find out what we have been using for treating SARS patients. Although the data is lacking from randomized clinical control trials, we do have some historical case control studies. And some of the examples include interferons and ribavirin, and also lopinavir, which is actually another protease inhibitor that you have mentioned in the treatment of HIV-infected patients. So we tested these drugs, and we found a number of interferon preparations, and we found that betaferon among them is the most potent in inhibiting MERS coronavirus. And to give you a perspective of how potent it is, the IC50, which is the 50% of the concentration of the drug that is needed, the concentration of the drug that needs to inhibit 50% of the virus infection in vitro, is around 17 international units per mil. Now, what could be achieved clinically as the peak serum concentration after subcutaneous dosage is about at least 2 times higher than this. So that makes it a possibility. And actually, betaferon or interferon beta 1b has been frequently used in patients with multiple sclerosis. So we would know quite well the PK/PD profiles and also side effect profiles.
And safety.
Yes,
You
exactly.
know whether or not it would be safe to
and
use.
the dosing regimen and so on. And then we also perform another program called drug repurposing program. And basically what we do is we use an automated robotic machine to screen a chemical library consisting of more than 1,000 drugs that is available commercially and is also FDA approved for other purposes, to see whether they also have anti-MERS coronavirus activities. And what we found is that mycophenolic acid, which is available usually as mycophenolic mofetil or mycophenolic sodium salt, and is used usually as an anti-rejection therapy in patients with organ transplantation, was highly effective. And when we compared mycophenolic acid with ribavirin, because ribavirin was another drug that was used in SARS, and also it's a very broad spectrum antiviral, we found that the IC50 to Cmax ratio is much better in the mycophenolic acid. So theoretically, if you calculate everything, you make the calculations, we show that mycophenolic acid only after a dose of 1 gram orally, which is not a very high dose, and it's clinically relevant, the concentration in the serum should theoretically last for at least 5 days higher than the IC50 for MERS. Now, but there's one particular concern about the use of mycophenolic acid in such a case, because mycophenolic acid has some immunosuppressive effects, right? Obviously,
I was going to ask you about that.
we don't want
sort of the double-edged sword.
So further animal studies should be confirmed to address this issue, but we believe if you can only give 1 small dose at the beginning of the MERS infection to the patient, and then you just stop it and you wait, because the dosage that gives the peak serum concentration to be above the IC50 should last for about 5 days already. And that should not have a very significant side effect as in organ transplant recipients who are regularly taking the drug. But again, everything I've said are postulations, so we have to confirm in further animal studies and hopefully clinical trials.
I suppose that's the good news, bad news piece for your story is SARS was blessedly short-lived as an epidemic, and then as a virus that we are concerned with, it seemed to burn out fairly quickly. But what your studies are showing is that work from Dr. Lee's group is very important.
It's very important.
And developing these new protease classes, because there are many more bats out there with many more coronaviruses, and perhaps we are going to set up a collaboration here from the couch where you can actually begin to test some of the compounds in her library to see whether or not they're active against some of these other coronaviruses. So as an emerging graduate student, what do you think of all this stuff about drug development and looking at crystallography and all of these things?
Nowadays, crystallography actually is a well-established method widely used in the pharmaceutical industry, in the academia. So I don't know if everybody is familiar with that or not. Basically, it's very easy. It's like you use microscope to observe the bacteria, but now we wanted to observe how it looks of the protein structure. But there actually is no such microscope can enlarge until you can see the structure of the protein. So we use X-ray to diffract the protein, and we collect the data, and we reconstruct the 3-dimensional image by the computer programs. So this is a very visually direct, can visualize like how the protein looks like, and if you're lucky, you get the co-crystal structure with your inhibitor with the protein. You can even get the information like how the inhibitors interact with the proteins. I think in the drug, not in the drug stage, but in the inhibitor optimization stages, like you want to expand the chemical scaffold, you want to build more interactions, enhance its binding affinity or inhibitor affinity. This relies a lot on the crystallography studies, because you can, based on the interaction patterns, you wanted to build more interactions based on the current structure. You can get the information from the crystal structure of the protein and your compound.
So, Dr. Lee, how did you get interested in proteases and protease inhibitors?
How did I?
Yeah.
Actually, our group have been working on multiple viruses. Actually, hepatitis C virus is one of my project too. I work on. And
hence the proteases.
Yes, that one too. So, like you mentioned, HIV protease, HCV protease, there are marketed drugs. So it was pretty safe.
Logical to make the leap.
Right. So since our lab is not dealing with the actual virus-infected cell, target-based drug discovery is our specialty. So I think there are good side on target-based drug discovery as well. So that's why we chose protease.
And so looking at the protease inhibitor, when you hear the discussion from Dr. Chan about these immunosuppressive drugs, it's quite attractive to think about using a cocktail so that you don't see emergence of resistance, because we know that you can select out protease inhibitor resistant virus simply because that what does not inactivate the virus makes it stronger. So clearly this approach of looking at formerly immunosuppressive drugs in concert with some of these new emerging targeted to the virus approaches are extremely important.
Right. So what was the question?
I was just making a comment. So I guess for you, what's next? I mean, you're working on MERS, you're working on Hep C, HIV. Are you sticking with the proteases?
At the moment, yes. I guess we could develop and move forward to cell-based, more, in general, antiviral drug development, but at the moment, actually, we are sticking to target-based. But I mean, everybody have different specialties, and in the end, all information gathered together to make.
Which is one of the attractive reasons you're at the InterScience conference on antimicrobial agents and chemotherapy. Dr. Chen, where do you think MERS is going to go? Do you think we're going to see the emergence of epidemic, or do you think it's going to go the way of SARS?
Right, so one of the key questions was the transmissibility between person to person. And from the data that we have been provided by the WHO and also the Ministry of Health from Saudi Arabia and the other countries, it seems that the transmissibility is not as high as that of SARS, which is a good thing. But the fatality rate is much higher. So in SARS, overall, the case fatality rate is about 10%. But for MERS, up to today, despite the increase in number of detection of asymptomatic or mild infections, we still see a case fatality rate of about 30%, so which is at least 3 times higher. So it remains to be seen whether the virus will further adapt to human transmission, which is of course very important. But from what we have gathered from yesterday's symposia, we heard that the Middle Eastern countries, the physicians and scientists there are very collaborative nowadays. And with the help of the other authorities like WHO personnel, the numbers have actually gone down in recent 2 months. Now, but one keynote is to see what will happen during next April to June. Because in the past 2 years, the number has shot up during that season because that was related to when the camels were breeding. And as you know, the epidemiological link with the dromedaries, the human cases would go up in those months. So we have to be very cautious in monitoring not only the human cases, but also the molecular evolution of the virus.
You've been watching ASM Live at ICAAC, and we've been visiting about the MERS-CoV virus that's emerging as a threat to the globe. And we've been lucky to have Dr. Jasper Chen from the University of Hong Kong, Dr. Lei from the University of Illinois at Chicago, and Dr. Lee, also of the University of Illinois at Chicago, talking with us about their strategy, literally moving from the bench to the bedside. Join me again in a few minutes for the next edition of ASM Live here at ICAAC. I'm Michael Schmidt from This Week in Microbiology. Feel free to join us again for another edition of TWIM when Moselio Schechter from Small Things Considered, Michelle Swanson from the University of Michigan, and Vince Racaniello, our host of TWIM from Columbia University, get together to discuss the unseen life on planet Earth. TWIM, the virology, about the microbiology that doesn't talk about virology, Vincent, about microbes. Thank you, everybody, and we'll see you again soon.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. anybody who has access to this facility first has to go through an R scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.